Transcription is the process by which genetic information encoded in DNA is copied into RNA. It occurs in both prokaryotic and eukaryotic organisms, but there are some differences in the transcription process between the two. In prokaryotes, such as bacteria, transcription takes place in the cytoplasm. The DNA molecule is unwound, and an enzyme called RNA polymerase binds to a specific region on the DNA called the promoter. RNA polymerase then moves along the DNA strand, synthesizing a complementary RNA molecule called messenger RNA mRNA. The mRNA molecule is synthesized in the 5:3 direction, complementary to the DNA template strand. Transcription continues until the RNA polymerase reaches a termination sequence, at which point it detaches from the DNA and releases the newly synthesized mRNA molecule. In eukaryotes, including plants and animals, transcription occurs in the nucleus. The DNA is also unwound, and RNA polymerase binds to a specific region on the DNA called the promoter. However, unlike in prokaryotes, eukaryotic DNA is more complex and contains non-coding regions called introns with encoding regions called exons. During transcription, the entire DNA sequence, including both introns and exons, is copied into a precursor mRNA molecule called primary transcript or pre-mRNA. Pre-mRNA undergoes post-transcriptional modifications, including RNA processing, to produce mature mRNA that can be transported out of the nucleus and translated into protein. RNA processing involves several steps. Capping, a modified guanine nucleotide called a 5-cap, is added to the beginning of the pre-mRNA molecule. This cap helps protect the mRNA from degradation and is important for mRNA stability and translation. Polyadenylation, a string of adenine nucleotides called a poly-A tail, is added to the end of the pre-mRNA molecule. This tail plays a role in mRNA stability and helps in the export of mRNA from the nucleus. Splicing, in eukaryotes, the introns within the pre-mRNA molecule are removed, and the exons are joined together to form a continuous coding sequence. This process is known as splicing and is carried out by a complex called the spliceosome. Splicing is crucial for generating different protein isoforms from a single gene and increasing the coding capacity of the genome. After RNA processing, the mature mRNA molecule is transported out of the nucleus and into the cytoplasm, where it can be translated by ribosomes to produce proteins. The processed mRNA molecule contains the necessary information in the form of codons, which are triplets of nucleotides, to specify the sequence of amino acids in a protein.